Hey everyone, my name is Rylan Stunkle and I'm a graduate student at Virginia Commonwealth University. And today I'm going to discuss how interactions between organismal traits and landscape characteristics influence the abundance of two taxa in riverine rock pools. So connectivity and patch size are important landscape characteristics that influence species abundances and diversity. These landscape characteristics are principal components of ecological theories such as island biogeography, metapopulation theory, and metacommunity theory. More connected habitats typically have higher dispersal and colonization rates, which leads to greater species persistence, gene flow, and movement of energy within a landscape. Likewise, patch size can affect population densities and species richness by influencing immigration rates, habitat selection, and extinction probabilities. Additionally, habitat area is associated with the amount of complexity and resources within a patch. Habitat fragmentation is often a contributing factor to declines in biodiversity, which really highlights our need to improve our understanding of how patch size and landscape connectivity influence population dynamics and shape patterns of diversity in terrestrial and aquatic habitats. But as you can imagine, differences in dispersal modes and abilities, as well as life histories, could lead to differences in how species respond to patch size and connectivity. For instance, organisms that fly will be less sensitive to low connectivity within the landscape than less mobile species that might move over land or through water. So these differences in responses to landscape connectivity and patch size can shape patterns of abundance and diversity both at the patch and the landscape scales. In ecosystems that are periodically connected by flowing water, such as riverine floodplains, species trait differences could drive changes in community structure over space and time. So this led us to ask the question, how do interactions between organismal traits and landscape characteristics influence the abundance of species in riverine landscapes? And to answer this question, we modeled the density of two focal taxa, the Virginia river snail and skimmer dragonfly nymphs, and compared their responses to hydrological connectivity and patch size in a system of riverine rock pools. We chose these two taxa because they have life histories and traits representing extremes of the hydrologic connectivity spectrum. For instance, snails are adapted to flowing streams and can only access rock pools when they are flooded. Conversely, dragonflies will be excluded from colonizing pools when they are inundated. Rock pools are an ideal system for addressing our question because they come in a range of sizes, they're easily sampled, and numerous in the landscape. Additionally, their hydrologic connectivity can be quantified, which I'll touch on later in this presentation. On the left, you can see a daily river heights from the USGS gauge upstream from our study site at Belle Isle. And on the right, you can see a map showing the number of pools flooded at the corresponding river height on the left. These pools vary in their height above and distance to the river channel, creating a gradient of flooding depending on the height of the river. And you can see in the figures how this gradient changes over space and time. We quantified the hydrologic connectivity of the system by modeling the height at which each pool floods corresponding to the river height at the upstream USGS gauge. We did this by capturing aerial images using a drone across a range of river heights. For each image we collected, we overlaid a shapefile of rock pool points and scored them as flooded or not flooded for a given river height. Then for each of the 750 pools in our system, we modeled the probability of the pool being flooded using logistic regression. The inflection point of the model is the pool's estimated flood height. We modeled the densities of snails and dragonfly nymphs as a function of patch size, connectivity, and season using generalized linear mixed models. The components of patch size we included in our model were pool surface area and mean depth. The elements of connectivity we included were pool distance to the river channel, the river height at which a pool floods, and the number of days since a pool was last flooded. The dragonflies we studied are seasonally migratory and their development is temperature dependent. so we included season in our models. We collected 523 samples from 302 pools over 3 years. We included pool ID and year as random effects in the models. Additionally, we scaled and centered our predictor variables so that their relative effect size could be compared within and between the two models. Because snails can only move to and between pools when they are flooded by the river, we expected snail densities to decrease as pool flood heights and distances to the river channel increase. We expected the opposite for dragonflies that primarily carry out the nymph stage of their life cycle in small lentic habitats. Additionally, we expected nymph densities to increase as pools were isolated from the river for longer periods of time. For both taxa, we expected increased resources associated with larger pool size to sustain higher densities following colonization. We found key differences in how these taxa respond to connectivity. Snail densities decreased with increasing pool flood height and distance to the river channel, while neither of these variables was a strong predictor for dragonfly densities. Nymph densities increased with the number of pool days a pool was isolated from the river, while there was no effect on snail densities. Only snail densities increased with pool surface area, and depth was a poor predictor of dragonfly and snail densities. As expected, dragonfly nymph densities were higher in fall and summer than in spring, while we found no substantial difference in snail densities among seasons. So given that we have found habitat connectivity is influenced by species traits interacting with environmental conditions, we might expect that at the landscape scale, greater biodiversity is maintained when patches experience varying levels of spatial and temporal connectivity. I would like to thank the people listed here. I'm happy to take any questions that you might have.